Good afternoon. My name is Rachel Best, and I'm a PhD candidate at Florida State University in Dr. Don Levitan's lab. And today I'm going to be talking to you about some of my dissertation research, which looked at the role of phenotypic plasticity as well as genotype by environment interaction in an asymbiotic species of Octocoral. So organisms have the ability to vary in their morphology, which can be due either to genetic differences or phenotypic plasticity. And we can think of the variation of phenotypic plasticity as the combination of both how the environment is driving differences in phenotype, as well as how genotype by environment interactions are causing phenotypic variation. And we can define genotype by environment interaction, or G by E interactions, as the variation between genotypes in their plastic response to the environment. And so phenotypic plasticity has the ability to buffer populations from temporal environmental variation, as well as help buy time in the event of environmental change. However, under variable, unpredictable environments, it can cause maladaptive plasticity. And G by E interactions can allow for selection to act on plasticity, which can potentially lead to the evolution of plasticity as a trait. So phenotypic plasticity has been looked at across depth in tropical mixotrophic octocoral species. So these are octocoral species that are obtaining their energy through both heterotrophically feeding as well as their photosynthetic symbionts. And so we've seen variation in the response by species, but some commonalities that we tend to see across depth are that colonies tend to be bushier in shallow environments, meaning that they have a higher density of branches. And they also tend to have a greater density of polyps in shallow environments. So as an example, this top photo here is showing a species of Antilogorgia in a shallow environment. So you can see the high density of branches as compared to this bottom photo, which is the same species, but located in a deep environment. And so phenotypic plasticity has been looked at in these mixotrophic tropical octocorals. But what about in species that do not photosynthesize? So across the globe, there are differences in distribution between octocoral species that have symbionts and those that are asymbiotic. And we expect that as you increase in depth, that you see a transition of species present going from predominantly species that do have symbionts to species that do not have symbionts. And so environmental variables that might lead to plasticity are things like water movement, temperature, salinity, and light availability, because they can have an effect on physical stress. They can increase the risk of colony detachment. They can influence food capture availability, and they can also have an effect on the photosynthetic potential of the symbionts. And so these differences in morphology that we've seen in previous studies on tropical mixotrophic octocorals varying by depth are often attributed to differences in light availability having an influence on the photosynthetic potential of their symbionts. But what happens in species that are asymbiotic, that don't have photosynthetic symbionts? How are they responding in their morphology to depth? And so I wanted to look at this using the asymbiotic octocoral Leptogorgia virgulata. This species is a predominant habitat builder on limestone reefs in the Gulf of Mexico, as well as along the east coast of the United States and the Atlantic. And this species can come in three predominant color morphs, shown here magenta, yellow, and orange. In a previous study that I conducted, I found that at inshore sites in the Northeastern Gulf of Mexico, we see magenta and yellow as well as orange color morphs present. However, on offshore sites, we tend to mostly only see magenta color morphs. However, doing some follow-up genetic analyses, I showed that colonies in these different locations, as well as these different color morphs, are likely the same species. But given this difference in distribution, for the sake of today's talk, I'm only going to be focusing on making comparisons between magenta colonies inshore and offshore to limit any potential morphological variation that might be driven by color morph. And so I wanted to study this in the Northeastern Gulf of Mexico, as the Gulf of Mexico is a notoriously highly variable environment. More specifically, in the Northeastern Gulf of Mexico by the Apalachicola Bay, where I conducted my study, we see even higher environmental variation because the Apalachicola River outputs right here, we see high fluctuations in temperature, salinity, chlorophyll A concentration, as well as turbidity. So anecdotally, this looks like sometimes you can see your buddy and the corals you're studying, and sometimes you're holding on to your buddy to make sure you don't lose them, and you're feeling around for those corals because you can't see them. So today I'm going to talk to you about two of my research objectives. First, I wanted to document whether or not there was morphological variation between these inshore and offshore sites. And then I wanted to see whether or not this species is exhibiting phenotypic plasticity of these traits and whether or not there are G/E interactions occurring. So to start off, I conducted a survey at 4 inshore and 4 offshore sites in the Northeastern Gulf of Mexico. The inshore sites averaged 3 meters deep on average, and were less than 1 kilometer from shore, whereas the offshore sites were roughly 13 meters deep and nearly 17 kilometers from shore. And so at each of these sites, I would run several transects, where along the transects, I would measure the maximum height of each colony, count the number of branches, and take a tissue sample for micromorphological analyses back in the lab, like polyp density. And so just to give you an idea of what these reefs look like, when you hear the word reef, you may be thinking coral reef, but these reefs are limestone reefs, which are predominantly comprised of octocorals, sponges, and macroalgae. So looking at polyp density, I found no significant difference between polyps at inshore and offshore colonies, which differed from what we've seen in previous studies of these mixotrophic octocorals, where they've had a higher density of polyps on inshore sites. We didn't see that in this species. Next, looking at bushiness, to orient you to this figure, along the x-axis is the height of the colony, and along the y-axis is the number of branches transformed using the natural log. So in general, we see that as you increase in height, you increase significantly in the number of branches that an individual has, which makes sense. As an individual grows, they increase in their number of branches. However, if you're located inshore, as shown here in green, as they increased in height, they increase significantly more in the number of branches than those located offshore, as shown here in orange. And so that's that difference in bushiness, where inshore sites have significantly bushier colonies than offshore sites, which is what we're seeing as well in those mixotrophic species. So colonies at inshore and offshore sites do vary in their morphology. We see significantly bushier colonies inshore than offshore, like they've seen in previous studies. However, there was no difference in polyp density. So then I wanted to look at whether or not this species is exhibiting phenotypic plasticity of these morphological traits. So as a reminder, if plasticity is not occurring, we expect that an individual is expressing the same phenotype regardless of the environment that it's in. However, if plasticity is occurring, we expect that an individual is altering its phenotype as the environment is changing. And so for this study, I wanted to look at the traits growth and branching. So to do this, I conducted a fully reciprocal study where I collected 8 colonies from 3 different sites. And I fragmented each of these 8 colonies into 9 fragments that were roughly 14 centimeters in length. And then I transplanted 3 fragments from each of these colonies for genetic replication back out to all 3 sites for a total of 288 fragments in this study. And so these 3 sites that I used differ in their depth and distance from shore. So site 1 is the closest to shore and the shallowest. Site 2 is slightly deeper and 2 kilometers from shore, and site 3 is the deepest at 14 meters deep and the furthest from shore at 20 kilometers away from shore. And so I transplanted these fragments by anchoring them to frames and then attaching them to the benthos as shown here. And what we see is that the site that they're transplanted to has a significant effect on the probability of whether or not they grew. So those transplanted to site 1 grew significantly more than those transplanted to sites 2 and 3, but where they came from, their origin, didn't have an effect on whether or not they grew. Of those that did grow, we see again a significant effect of where they were transplanted to, with those being transplanted to site 1 growing more than those transplanted to sites 2 and 3. But with this, we see an effect of origin as well as an interaction. And so we're seeing some of those carryover effects of where colonies originated. Looking at the probability of branching, we see a similar trend where those that were transplanted to site 1 branch significantly more than those transplanted to sites 2 and 3. Now, you may be thinking that these error bars are very large, and they are. And that is because the study was only able to run for 70 days due to an impending hurricane. So there wasn't a whole lot of time for these colonies to start branching. So it's really exciting that we are seeing branching in such a short period of time. So on the right is an example of this. So the top photo is a fragment coming from the same colony as the bottom photo, but the top one was transplanted to site 1, and you can see it has several branches, whereas the bottom photo is a fragment that was transplanted to site 3. And as you see, it's not branching. Another exciting thing is that when we're comparing to the natural average number of branches at each of these sites, we see a similar pattern of site 2 having a trend of a higher average number of branches compared to the other 2 sites. And we see this in our transplant experiment as well, which is really exciting. And so we are seeing plasticity of growth and branching. There is an effect of origin on the amount of growth. And we are seeing branching patterns that are mimicking the naturally occurring patterns of branching at these sites. So then I wanted to look at whether or not there are G/E interactions occurring. And so as a reminder, if G/E interactions are occurring with plasticity, we expect that different genotypes are responding to the environment differently. So for this, I'm only going to talk about my results for growth, given that the study was so short and had limiting amounts of branching. And so to orient you to this figure, along the x-axis is a pairwise comparison of two transplant sites. And along the y-axis, we see the average amount of growth. So in this first figure, comparing sites 1 and 2, we see a significant genotype by environment interaction occurring. Similarly, when comparing sites 1 and 3, we see, again, a significant genotype by environment interaction. However, when comparing colonies transplanted between sites 2 and 3, we're not seeing this genotype by environment interaction. And this is likely due to the small difference in plasticity or plastic response that we saw in colonies transplanted to sites 2 and 3, as compared to the significant growth we're seeing at site 1. So to wrap up, we see these differences in growth and branching in this system, yet with this species, they're likely unrelated to sunlight, even though they match some of the similar patterns that we're seeing in tropical mixotrophic species. We also see evidence of phenotypic plasticity that might be able to explain some of the naturally occurring variation in colonies on these reefs. I found genotype by environment interactions present for growth, which may lead to the possibility for the evolution of growth plasticity. And it's really exciting that we're seeing plasticity in this species because it does have that ability to buffer populations from temporal environmental variation as well as long-term change. And given that this species is such a critical habitat builder, it's exciting to see that they do have the capacity to respond to environmental change. And so with that, I would like to thank my lab, all of my field help, especially from undergraduates to graduate student peers to lab techs, as well as my funding sources. Thank you.